This video will explain Learning Outcomes 3 and 4, explaining a genomic test result and knowing when to contact genetic services. So let's explore how to explain a genomic test result further. There may be 4 possible results of a genomic test. The first may be a genetic cause has been identified that explains the clinical presentation. Alternatively, a variant of uncertain significance may have been found, or an incidental finding may have also been demonstrated. The last possibility is a negative result. To help manage patient expectations, it's important to discuss possible results with the patient and parents when obtaining genomic consent. In this scenario, Oliver's trio exome has detected a genetic cause. This occurs in about 30 to 40% of children presenting with global developmental delay or intellectual disability. A de novo, or new change in Oliver, has been demonstrated in the gene AHDC1. This allows for accurate genetic diagnosis in terms of management and counselling, restores reproductive confidence for the family if they wish to have further children, and enables access to literature and possible further studies, and in some cases, therapies. In this alternative scenario, Oliver's whole exome sequencing results report a variant of uncertain significance. This occurs in 20% of cases. It is important in this situation to explore the clinical presentation or phenotype of the child to see if the change in the specific gene fits with the clinical presentation reported in the literature with the gene. For further clarification, if the genomic test ordered was a singleton exome, parental testing may be required. If the change is inherited from a well and healthy neurotypical parent, it is less likely to be significant. A Medicare item number is in place for familial testing. Exploration of a variant of uncertain significance may require additional clinical and molecular correlation, counselling, and often research to clarify the implication. This is best done by contacting your local genetic service. For families, a variant of uncertain significance may increase anxiety and uncertainty. It is also often associated with increased time and cost. To explain the concept of variant of uncertain significance, the analogy of the spelling of the word harbour can be used. Harbour can be spelt with a U in the Australian English language version or without a U in the American English language version. However, an X in the word harbour is definitely a spelling change. A VU is where we cannot tell the difference between a normal variable way to spell the DNA sequence in a gene and a mistakenly spelt DNA sequence of the gene. Another scenario may be that Oliver's whole exome sequencing results report an incidental finding. An incidental finding may arise in about 1 to 2% of cases. This means a change is found in a gene that is not related to the clinical presentation or indication for ordering the sequencing, but may still be of clinical importance. Most labs do not actively examine for unrelated genes. Such a finding does not affect health insurance or other type of insurances that are already in place. However, new insurance applications for income protection, life or mortgage insurance may require mandatory disclosure at some insurance companies. This is more of an issue if an incidental finding is found in the parent of a child and therefore may arise when a trio exome is ordered. That is, ordering of exomes of child and both biological parents. This may require referral to genetic services to further discuss with the parents and family. The fourth possible outcome may be a negative result. This occurs in approximately 40% of cases. The possible reasons may be that the etiology is non-genetic, or as only 5,000 of our 20,000 genes have been related back to human disease to date, it could be that the gene is yet undiscovered, or that the methodology used to do the genomic testing is not advanced enough to detect the causative genetic variant. That is why reanalysis is recommended every 2 years and is covered by a Medicare item number. Further explanations in the event of a negative genomic test. There may be a tiny deletion or duplication that has not been detected by chromosome microarray. Alternatively, if the causative gene is mitochondrially encoded, it is important to realize that the mitochondrial genome is not examined when a whole exome is done, but is examined when a whole genome is done. Other explanations could be that there may be a methylation abnormality, such as Angelman syndrome, or that the genetic mutation is an expansion disorder, such as Huntington disease or Fragile X. Both these genetic mechanisms are not examined by most genomic tests to date. Lastly, unless a whole genome is done, a whole exome will not detect non-coding protein changes, that is, intronic changes. Additional reanalysis is covered by a Medicare item number at an interval of 2 years. Gene discovery and improved methodology may uncover these genetic causes with time. Now to learning objective number 4, knowing when to contact genetic services for assistance. Refer to discuss with genetic services when considering genomic testing and at any stage through the process, particularly if there are any concerns regarding the following: pregnancy, consent issues, how to order the test, interpretation of results, and discussion or exploration of a genetic diagnosis. Find your local genetic service at www.genetics.edu.au. This genomic testing pathway brings together the various steps involved in genomic testing, recognizing when genomic testing would be suitable for a patient, obtaining a genomic consent and ordering genomic testing on a patient, explaining a genomic test result, and knowing when to contact genetic services for assistance. It is important to remember that when you are considering genomic testing, under the Medicare item rebate, consultation with your local clinical genetic services is mandatory. Take-home messages in relation to the learning outcomes include: firstly, recognition when genomic testing is suitable. Any child less than 10 years of age with dysmorphism and/or one or more congenital structural anomaly or moderate intellectual disability, global developmental delay. This is following normal first-tier testing, which encompasses chromosome microarray, Fragile X, and urine metabolic screen. In addition, discussion with the family to make sure they are happy to proceed to genomic testing is critical. 2. Consent and ordering of genomic testing. Points to remember are: trio whole exome sequencing is the test of choice for intellectual disability and neurodevelopmental disorders. Secondly, consent for the patient only, singleton, or trio, that is patient and both biological parents, is mandatory. Thirdly, ordering of the test requires 2 to 10 mL of EDTA done at a selected lab for DNA extraction and storage. Consent for the child, that's a singleton, plus each parent if it's a trio. Requests need to have Medicare required clinical information plus any other relevant clinical features, and each lab will have its own phenotype form. 3. Explaining a genomic test result means that there are 4 possible outcomes: that a genetic cause is identified, or that a variant of uncertain significance is identified, or there may be an incidental finding, or that the test is negative, and this qualifies for reanalysis in 18 months to 2 years' time. Fourthly, knowing when to contact your local genetic service. They can be contacted at any time. Details for contact are at www.genetics.edu.au. We've looked at the genomic testing pathway, and as mentioned, it is important to know you can refer or discuss with genetics at any stage. Search the Centre for Genetics Education New South Wales Health website, www.genetics.edu.au, for details of genetics clinics in your area, and have a look at the available resources like the genomics information, genomic consent videos, and life insurance fact sheets. Frequently asked questions. What about autism? Single common genetic etiologies in autism spectrum disorder remain rare and a work in progress. It's important to exclude aneuploidy with chromosome microarray and also do Fragile X DNA testing. Single gene causes are rare. Single gene causes usually are associated with other comorbidities or clinical features such as moderate to severe intellectual disability, significant macrocephaly, dysmorphism, epilepsy, involvement of other organ systems, and/or family history of autism or intellectual disability. Other frequently asked questions. Will the Medicare item number cover all clinical presentations, example hearing loss, connective tissue disorders? Even if the specific criteria for these item numbers are not met, many conditions warrant genomic testing. Please refer to your local genetic service. Will I have to contact a clinical geneticist or clinical genetic service every time I want to order a genomic test? Yes, this testing must be done in consultation with a clinical geneticist. Please follow your pathway with your local genetic service. Whole exome sequencing versus whole genome sequencing. Should we do whole genome sequencing since it's better? This is laboratory dependent. Please discuss with your local genetic service. What is the turnaround time for a result? This is laboratory dependent, but the average is about 3 to 6 months. There are many resources to help prepare for genomic testing, and these are listed on this slide. Thank you for listening to this video.